All right. Are you ready? Let's strap in. Transcription and RNA processing. This is a wicked cool topic. You're going to love this. So, think all the way back to Unit 2, proteins. Proteins are polypeptides. Poly, meaning more than one. Peptides refers to the bond. They're made up of amino acids. String of amino acids is a polypeptide. Poly, meaning more than one, peptide is referring to their peptide bonds. So amino acids are linked by peptide bonds. Proteins are polypeptides made of amino acids. Amino acids are bound by peptide bonds. It's a tongue twister. When we talk about gene expression, what we're really talking about is this process by which DNA directs the synthesis of a protein. So it directly goes from DNA to RNA to proteins. So this includes two phases, and RNA is smack in the middle of it. We have transcription, which is where we are taking the DNA code and transcribing it to RNA. And then we have translation that will take that RNA, that messenger in the middle, and translate the message into a protein, which is then expressed in the body in some way, or in the cell in some way. This occurs everywhere, all organisms. This is everything. DNA to RNA to protein. Pause here if you got to stop and take notes. So let's break this down. Transcription, translation. Transcription, what we really mean by it, it's the synthesis of an RNA molecule using information from the DNA. It allows for the message of DNA to be transcribed, right? Written, transcribed from one to the next, and it occurs exclusively in the nucleus. All right, you can see it right over here in the nucleus. Think of the DNA as being the ones and zeros in a computer program, right? So your computer runs on ones and zeros. I don't know exactly how it works, but those ones and zeros are then transcribed into a number sequence that is then translated into something your computer does, right? So it's a code that's being coded and then decoded and then decoded. Translation is the synthesis of a polypeptide. So it's the synthesis of a protein using the information from the RNA. So the RNA gets its information from the DNA. This occurs on a ribosome. Unit 2, ribosome, cytoprotein synthesis. This is the protein synthesis we're talking about. A nucleotide sequence, it's essentially, this is like the end all and be all of it, right? Is the nucleotide sequence becomes an amino acid sequence. So the A's and the T's and the C's and the G's become A's and U's and C's and G's. Then they become this nucleotide sequence that we'll get into, this amino acid sequence that we'll get into. Before we get started, we have to talk about pause here if you have to continue taking notes. All right, so before we get into transcription translation, we got to talk about a couple different key RNA molecules. The first is messenger RNA. We've heard of this one throughout COVID, mRNA molecule. The M stands for messenger. The second one here, which is one of my favorites, is the pirate RNA. It's the rRNA. R stands for ribosomal RNA. The ribosomes are an organelle that are actually made of protein subunits. So we need an RNA molecule to make them. It's the rRNA. And the third is the tRNA. It's the transfer RNA. And that RNA will actually transfer the amino acids over to the polypeptide sequence. Pause here if you need to finish taking notes. All right. Messenger RNA. This one's, they're all important, but this one is the thing that kicks everything off. So messenger RNA is synthesized during transcription using a DNA template. So if you think back to last topic when we talked about DNA replication, the same kind of process happens here where you have a replication fork here, a replication fork here. This is still going in the 3 to 5 prime direction, except instead of having another strand come up here, we have just the single RNA strand that you see, and that is characterized by the U that you'll see there. Cool. Ooh, that's a fun thing. So the mechanisms are similar. We still got the helicase and the topoisomerase and all that great stuff. It's just a different molecule being made. mRNA will then carry this information from the DNA inside the nucleus to the ribosome. So they're sending the message. They're the messengers. Pause here if you got to take notes. Cool. Let's talk about transfer RNA. This is the second type of RNA that we have to speak about. So transfer RNA are, they're molecules. Yeah, obviously they're important in the process of translation. Essentially what happens here is you have a piece of RNA over here that can carry an amino acid, a specific amino acid, to the codon that we'll see on the mRNA. On the mRNA molecule, we call it the anticodon, complementary codon to the mRNA over here. So allows for information to be translated. All right, let's break this down. So amino acid sequence, a specific amino acid will be hanging out up here. Ooh, I like this. Hanging out up there. And then you will see here we have the anticodon. In this case, it's GAA, which will correspond to the codon on the mRNA that is CUU. Right? So when we look at reading frames, when we talk about codons and anticodons, this is really what we're talking about. These loops over here, don't worry so much about it. These are just kind of the weird sort of offspring of the tRNA to begin with. Go ahead. If you got to take notes, pause here. Oh, there you go. Anticodon. Boom. Yeah, right there. That's a big one. That's the one that you got to know. Anticodon there. Various amino acids up there. Whatever the recipe calls for. Good stuff. All right, finally, we got to talk about ribosomal RNA. So our RNA helps form ribosomes. Derp, it's in the name. It essentially helps link amino acids together. And for the purposes of this class, that's really all you got to know about it. It's got a smaller part up here and a larger part down there. You know, I'm just showing you that because I want to show off the fact that I learned how to mark these up. Cool. So ribosomal RNA, it just makes ribosomes. Ribosomes are a protein, and we need an RNA to make the protein that makes the proteins. Cool. Pause here. So, talking about this coding. How do we decode? What is this code? Is this the Da Vinci Code? No, it's the genetic code. So DNA, as we know as scientists and from the first topic here and from Unit 1 and 2, DNA contains a sequence of nucleotides that codes for proteins. The sequence is read in a group of 3. We call this a triplet code. And this allows us to do some pretty interesting things that we're going to see. During transcription, only one DNA strand is being transcribed, so we call that the template strand, right? Sometimes it's called the non-coding strand. I've literally never heard it called the minus strand. I think that's a little mean. You know, it's got feelings. It doesn't, but you know, you get it. Antisense strand, I have heard before, but here we're going to call it the template strand. It is the template that we are using to build our, I can change the color. It's the template that we are using to build our protein out of. So pause here if you need to take notes. All right. So you can see here, we start with the DNA template strand. mRNA molecules are formed anti-parallel. Remember, there's directionality in DNA and RNA. Things go this way when they're being transcribed, right? Codons are read the same way. There's directionality in this. The strands are anti-parallel to each other, meaning you have a 3' end that matches with a 5' end and a 5' end that matches with a 3' end. As long as they're mirrored for you, as long as it's opposite, you are good. And they're complementary to the DNA nucleotides. So you see A goes with U, T goes with A, C goes with G, G goes with C, so on and so forth. No surprise here. Base pairing AU and CG. Savage, stop it. I'm sorry. Yeah, there you go. Better. All right. A pairs with U, C pairs with G. The nucleotide triplets, we call them codons. So, sorry, my cat's food's coming out. There you go. So that's one codon. It's another codon. And the codons themselves code for each amino acid, right? So there's a direct link here between DNA to mRNA down over to protein. This is how the whole thing's decoded. Pause here if you need to take notes. Oh, there you go. Yeah, look at all those arrows. Very nice. Like, duh. Yeah, like we see them. All right, whatever. There you go. Amino acids. All right, so triplet code. It allows for some really cool things here. So there's 64 different codon combinations, 61 code for amino acids. The other 3 are stop codons. They are universal to all life. Everything from me to my 3 cats that are currently eating dinner, to the plant that's hanging from my ceiling, to you guys listening to this here, all life has these codons. So talking about, and so there's 3 stop codons, and you'll see them up here. That's UGA. Do they not have the others here? No, just kidding. They're probably like an AUG. Where's an AUG over here? That's methionine. All right, so AUG over here is a start codon. And it's actually, oh, here we go. There's 2 more. There's the other 2. UAA, UAG, UGA, those are stop codons. They tell things to stop. AUG is methionine, also a start codon. We'll talk about that when we get into reading frames, etc. So what's really cool is the redundancy. This is what we call the redundancy in the genetic code, is that more than one codon can code for each amino acid. So even though there's only, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 23 different amino acids, we have 64 different codons. So what this means is that the DNA and the mRNA molecules can be more complicated. It's a more complicated code that we are simplifying. Is that the last there? Yeah. Ooh, it stays on the screen. How fun. This also allows for a little bit of leniency when it comes to mutations. So for example, if we had a point mutation in any of these codons, right? So let's say we have a point mutation where this uracil turns into, an adenine. It's still going to code for this amino acid here, which kind of gives the DNA a little bit of leeway if so to speak, if there's a random mutation of some kind. All right, stop and start codons. What the heck are they about? So there's a reading frame to RNA molecules. So the RNA molecules must be read in the correct groupings during translation to synthesize the right proteins, right? So for example, Mayhem, this isn't a call out to you. I'm not calling you out. The fat cat ate the rat. Cool, that's fine. We can all read that. The reading frame, if the reading frame shifts, right, or you, I don't know, add an extra one in there, extra letter in there, or anything to screw up that sentence, it turns into this, right? All we did here was remove the T, and you can see the T at the beginning of this, and all of a sudden this sentence made of 3-letter words, it makes no sense at all, right? So we have to have a reading frame. A place to start is really what it is. And that place to start, I should say, is AUG. It will always be AUG, AUG for life. Now, if there is a start codon, yes, start codon here. Look how happy it is. It's so happy. It looks kind of evil. All right, start codon here, and then dump, bump, bump, and you see an AUG before you see a stop codon. That is the amino acid methionine. So it is a start codon, but it's also codes for a specific amino acid. Anyway, a little bit of a tangent before we get there. The only thing you got to know for this is the reading frame. If you screw up the reading frame, everything gets screwed up. No more protein, no more organism. Too bad, so sad. Pause here if you got to take more notes. All right, quick check. If the DNA template strand reads 3' ACG AGA 5', what will the mRNA transcript read? Pause it, write it down. Yeah, you're right. 5' UGC UCU 3'. Lovely. Please remember to put your primes and understand that your 3' matches with your 5', and not the other way around. Using the mRNA transcript from number 1, this one up here, what amino acids will be produced during translation? You may got to go back and take a look at that codon chart if you haven't memorized it already. No, I'm not going to make you memorize that. That's insane. Look at the chart, write it down. Yup, cysteine and serine. You guys are so smart. I love it. Okay, let's break down transcription. This is the first step in this whole idea of making a protein. There are 3 steps to transcription. Step 1 is initiation. We first have to initiate it. We have to start it. It's the initiation process. Next, we got to make it longer. Got to elongate it. Lastly, we got to terminate it. We cut it off. So initiation, elongation, and termination. We're going to go over each one individually. Step 1, initiation. So this is going to look a lot similar to DNA replication from the last topic. So transcription begins when RNA polymerase molecules attach to a promoter region of DNA. So not a primer, but a promoter region. Does not need a primer to attach. No primers, yes promoters. The promoter regions are upstream of the desired genes, right? So remember, things get transcribed from 5' to 3'. So promoter regions are upstream of this. You'll see that on the next slide. Go ahead and pause here to take notes if you have to. All right. So, in eukaryotes, the promoter region is something called the Tata box. Tata. I'm sorry, when I drew upstream over here, I drew it wrong. 5, 2, 3. If things are flowing down that way, then upstream is up here. So it's past the 5 prime. Sorry about that. Just wanted to correct myself. So upstream is prior to where the actual transcription is going to begin. All right. Now that I've corrected myself sufficiently. In eukaryotes, the promoter region, we call it the Tata box. Yes. Get out your giggles. There's something called tatas. But what it really is it's just a series of T's and A's and T's and A's. And this whole region's the promoter region. So this whole thing would be tatas. And things called transcription factors will come and bind. Don't worry about an actual name. They're just factors that help transcription. They're not anything crazy. This all happens in an RNA polymerase, right? So the RNA polymerase itself binds to this promoter region. We call it the Tata box, and it brings along with it other transcription factors. This is done upstream of the transcription start site. Prokaryotes are a little bit different. They're simpler. They're easier. RNA polymerase can directly bind to the promoter. They don't need these transcription factors, the factors that help in transcription. They don't need any of that nonsense. They're independent. They can do what they want. They could do what they want. So promoter sequence is this whole sort of thing here. RNA polymerase, boom, comes and binds onto it, holds it tight, still upstream of the transcription start site. And when we say upstream, we mean before the actual gene that's going to be transcribed going that way. Woo, good stuff. Oh, there you go. Notice that the promoter is always upstream of the gene in interest to be transcribed. So yes, this is downstream. Imagine the mountains up here. And there's a waterfall coming down. And that's downstream. Downstream. Maybe I'm just reminding myself. All right. Pause here if you got to finish taking notes. Step 2, making it longer. Elongation. So RNA polymerase opens the DNA, reads the triplet code of the template strand. Lovely. We move this, so polymerase moves in the 3 to 5 prime direction. It elongates in the 5 to 3 prime direction. So 5 prime you can see down here, 3 primes up here. It's adding them in the 5 prime to 3 prime direction, but this is moving on the DNA in the 3 prime to 5 prime direction. So things move this way, 3 to 5, things grow 5 to 3. Moves one way, 3 to 5, grows 5 to 3. Ooh, sorry, wrong button. And that is true for replication as well. So things move 3 to 5, but are added 5 to 3. Pause here if you got to take notes. Oop, here. Cool. But it's not that simple. Polymerase moves downstream. It only opens small sections of DNA at a time. This is not replication. We don't have to go through the whole genome to be able to make another copy. we're only looking at the segments of DNA that actually code for stuff. So it only opens small segments at a time, and there you go, RNA polymerase pairs on complementary DNA nucleotides. Just one at a time, just pops them on. Growing our mRNA strand will literally peel away from the template DNA strand. This has been observed under microscope as well. So I'll see if I can find a video of that. It's pretty cool. Double helix then reforms. DNA just goes back to where it was. Helicase goes away. Topoisomerase goes away. Polymerase goes away. Everything's fine. If there's a long DNA, or I should say a long gene sequence, it can be transcribed simultaneously by several polymerases. So you'll see the DNAs going through, and then you have a polymerase here and a polymerase here, and they're kind of working, their way from one to the next to actually transcribe the whole thing, right? That's if a that's if a gene is very long. This helps increase the amount of mRNA synthesized, especially if you need, like a ton of mRNA, like if there's an immune response or something. You need a lot of protein really fast. Increases protein production. More mRNA equals more proteins. Pause here if you need to take notes. Finally, termination. So let's get this over with. Termination, we're done. We're done mRNA molecule. So in prokaryotes, it's a little bit different. Transcription proceeds through what we call a termination sequence. So once it'll hit this sequence of DNA, it causes the termination of a signal. So it causes the whole thing to stop. RNA polymerase will then detach. mRNA transcript is released. And then it goes out and proceeds to translation. This is in prokaryotes. Remember, prokaryotes don't have a nuclear envelope, so they don't have to move out of the nucleus. They are in just a nuclear region of their cell over here. So it just goes right into translation. This is for prokaryotes. mRNA doesn't need any modifications. It can just go and do its thing. It is fine. Eukaryotes, however, pause here to take notes. Eukaryotes, however, we're a little more complicated. We have that nuclear envelope. So RNA polymerase transcribes a sequence of DNA called the polyadenylation signal sequence. We call it a poly-A tail. And it codes for this sequence here: AUAAA, 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 AUAAA. Once they see that, RNA polymerase will release. Everything's good. So this releases the pre-RNA from the DNA. So when the RNA molecule is done being transcribed from the DNA molecule, it's just a rough cut. It's a rough director's cut. We call this pre-mRNA. We haven't made the necessary changes yet to make this legit. It's got to undergo modifications before translation. Yeah, we got to put all this stuff through editing. Got to make sure that everything's okay. So let's talk about pre-mRNA modifications. So there are 3 modifications that must occur to eukaryotic pre-mRNA before it's ready for translation. Number 1, a 5' cap. On that 5' end, we got to cap it. 2 is a poly-A tail. A-U-A-A-A, A-U-A-A-A. Finally, it's RNA splicing. Go splice it up. Pause here if you got to take notes. Awesome. So the 5' cap, it's on derp. It's on the 5' end. 5' end, 5' cap. It receives what we call modified guanine cap. It's just a bunch of guanines. G, 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 G. Bunch of G's. The 3' end gets this poly-A tail. We call it a poly-A tail. Just a whole bunch of A's. A. Poly meaning more than one, A meaning adenine. So it's just a bunch of adenines. It's different than the stop sequence or the termination sequence of AUAAA. This is something that happens after. I know I was chanting that. This is something that happens after the mRNA has been released from you're released from the DNA before it leaves the nucleus. So again, poly A tail, just a bunch of A's. A. 50 to 250 of them. Both the 5' cap and the poly A tail help to do these things. It helps it leave the nucleus, essentially. That's a big one. It's got to squeeze its way through these nuclear pores, so we got to make sure that it's protected. It protects it from degradation, right? If once this mRNA molecule is transcribed into a protein, we don't need it anymore. The cell doesn't need it anymore. No one needs it anymore. So it degrades over time naturally. That's just what our body does. Lysosomes come in, that sort of thing, and degrade it down. So this poly A tail and the 5' cap, it helps to protect it from actually doing that before it turns into a protein. It also helps ribosomes attach to the 5' end of the mRNA, right? It's going to have to attach somewhere. So let's have it attach here. Why not? It's upstream. So yeah. So those are the three benefits of these modifications of the 5' cap and the poly A tail. Pause here if you got to take notes. All right. This is cool. When I was taking this class in high school, we had RNA splicing. RNA splicing was the thing. So we knew that sections of pre-RNA called introns are removed, and then exons join together. Introns don't, so how I remembered this is introns stay inside. Introns stay inside the nucleus. They're not going to go make a protein. The exons exit the nucleus to go on to make proteins. Introns, intervening sequence, they don't code for anything. Exons are the ones that code. So the exons will be exiting, the introns will be staying in. You can see here, there's that 5' cap, there's that poly A tail, keeping it nice and protected. Pause here if you got to take notes. What we didn't have when I was in high school taking this class was alternate splicing, which is what this is. This was discovered sometime after I took AP Bio. So alternative splicing is a way that we can take one piece of DNA or one gene and code it for more than one kind of polypeptide. So if we follow these exons here, these are the ones that are going to be exiting the cell, and we take exon 1 and we pair it with 2, 3, 4, and 5, it's going to make this protein. But if we take the same exons, 1, 2, forget about 3, 4, and 5, we make an entirely different protein. How cool is this? This is something that wasn't discovered when I was taking this class in ancient times. So we take one piece of DNA, heck, it's even the same exons. We're just mixing them up different. We're just putting 1 and then 3 and then 5 and then it makes something crazy. Isn't that wild? I think that's so fascinating. So this is called alternative splicing. And this is another way that we can make every protein under the sun with just with just A's and T's and C's and G's, if we're being honest. Like, that's how it boils down. And it's because of this alternative splicing. It just blows my mind. It's so cool. Go ahead. Pause here if you need to take notes. Or just catch your breath, because man, that was a cool concept. Whoa. All right. Once everything's done, we have a mature RNA. It can finally leave the nucleus. It's going to fly the nest. And it's going to proceed to the cytoplasm for the translation at ribosomes, because ribosomes are the site of protein synthesis. Quick check. Discuss the overall big picture of transcription. What's transcription all about? What's going on? Pause the video. Think about it. Write it down. When you've written it, unpause the video. You're right. Transcription is this process that it writes down the information from DNA and allows it to leave the nucleus. It does that through mRNA. That's the whole kind of overall picture of transcription, is we're trying to get this DNA into a language that the ribosomes can translate, and we're trying to pass it through difficult terrain without damaging it. That's the big picture. How does the initiation differ between eukaryotes and prokaryotes? Go ahead, take a minute. Pause the video. Write it down. There's a couple differences. Big one being eukaryotes require transcription factors. Without transcription factors, RNA polymerase cannot bind. Therefore, mRNA cannot be produced. Therefore, neither could proteins, right? Prokaryotes don't need those transcription factors, the factors that help transcription. They're just factors. Don't worry about it. That help it bind, right? But prokaryotes don't need that. They can bind on their own. That's it. Congrats. I'll see you in class tomorrow. What? Discard. Cool.